Morning students. Today I am going to take a small revision topic, complement. Okay, before explaining about the complement, I want to stress one point here. Complement spelling, it is not P-L-I-M-E-N-T, it is P-L-E. Complement, what we give, what we get is different, and complement which is present in the blood component, it is different. So remember, the letter E is important in the spelling of complement. You know about the complement, these are the proteins which are present in the blood. There are about several complement proteins which are present, like C1, C2, C3 till C9, and also certain factors like factor B, factor D and other regulatory proteins are also present. These are all includes under complement. The basic character of the complement is it lies inactive when in the blood. It gets activated by some either antigen antibody complex. This is particularly the complement gets fixed with the antigen antibody complex, particularly the Fc portion of the antibody it goes and attaches to, right? So this all things, what is the function of the complement. The important things in the complement to be remembered is, one is pathways, other is regulation and deficiencies. So I'll be stressing on only these topics. Remaining things is already, no need to discuss again and again. I'm going to discuss what to be known, and you have to learn this, that is pathways. So we have three types of pathways, classical pathway, alternate pathway, lectin pathway. So a common thing in all this is they are making ultimately membrane attack complexes, which makes the cell die by making holes. So whatever the pathway it is, the action is the same, but how these pathways differ is basically the initiation step. The classical pathway is initiated by antigen antibody complex, whereas alternate pathway and lectin pathway are initiated alternate by antigen and lectin by antibody, right? So this, if you remember, you can write which belongs to innate immunity and which belongs to acquired immunity. The classical pathway belongs to acquired immunity because antigen is formed, antibody is attached, antigen is attached to the antibody, so it comes under in acquired immunity. And alternate pathway and lectin pathway, still no antigen antibody, no initiation of the response, so these all included under innate response. So these things, let us see directly the pathways. What are the key steps which are present in the pathways? Usually the complements will be inactive, as said. They should be activated. What are these complements? These are only proteins. How do we make the proteins activated? By lysing them. That is, we need proteases. Protease is breaking the protein to get two fragments. What are the fragments which we get in the complement? From C we get either 1, 2, 3 or A fragment and B fragment. Which is active in A and B? B is active and gets through the pathway, and A will be coming out. That you can remember. That if you can remember this simple tips, what is needed, then you can write the complete pathway. So there are complements C1 to C9 and also factors also present. The complement get activated when they are broken, proteolyzed to C3A and B. Is not only 3, N, A, B. Only one case. Before going to that, what is the initiation? As I said, initiation for the classical pathway is antigen antibody complex, and the alternate pathway is either one is antigen, another is lectin pathway is by antibody, and the initiation is different in different pathways. And the formation of C3 convertase is the key step in the pathways. Once the C3 convertase is formed, remaining steps are all similar, because once C3 convertase is formed, we can add 3B to the C3 convertase to get the C5 convertase, and all will form the membrane attack complex, which is similar in all the pathways. Let us see the pathways. As I said, the classical pathway is started by antigen antibody complex, and these are initiated by different things, either by antigen or lectin pathway by antibody. Here how they are get initiated we will discuss in each one. And what is the common thing in all these is the membrane attack complex. What is it made up of? It is made up of five components, that is 5, 6, 7, 8, 9. In the 5, the active component 5b is present. So membrane attack complex is common. Which is the membrane attack complex? It is 5b, 6, 7, 8, 9. So once you got the membrane attack complex, you know our ultimate goal is to get this membrane attack complex through any of these pathways. And what is the key step? I said it is the C3 convertase. Any pathway has to come and change the C3 into C3b and C3a. The C3 conversion is important key step for all the pathways. Once it is converted, remaining all are similar. So what is the necessary enzyme for C3 conversion is different in different pathways. First we'll see the classical pathway, because it is a key one. Remaining all depends on the other things. How the classical pathway is activated? As I said, for all the components, C components, A will be the inactive form and B is the active form. But whereas C1, there are 3 parts. In all 2 parts are present, but C1 there are 3 parts, Q, R, S. This Q, R, S, the Q is the major binding part. It contains 5 lobes, so that it can bind to 5 antigen antibody complexes if it is IgG. If it is IgM, only one can be fixed to IgM, because you know IgM is pentameric. So C1q is the major part which binds to the antibody, and C1r and S are required for conversion of C4. So the initiation for classical pathway is antibody complex, where C1q is binding one and C1r and S changes C4 to C4a. Actually the number is almost 1, 2, 3, 4. In that way only we get the numbers in the complement, except 4 which jumps to the first position. Only C4 jumps again. The C2, 3, 5, 6, 7, 8, 9 is everything is in order. So 4 is jumping to front. It wants to come first. So the first one is C4. After C1 is C4. C4 which is binding the C4b is the active site, and the C4a which is released, which acts as an anaphylactoxin or the opsonin, you know that. And one more point to be remembered is in C2 only C2a is the active part, not the B. B is released and it has a kinin-like activity. So 3 points to be remembered in the proteins. C1 has 3 sites, Q or S. C4 jumps forwards, comes after C1, and C2 has 2A as the active site. That is how, if you remember these 3 points, you can write about all the complements in order. The pathway can be completed easily. So what is the C3 convertase in the classical pathway? First is the C1, next 4 is coming front, so becomes C4b active component. And what is the active component for 2? It is 2a. So the C3 convertase for the classical pathway is C1, 4b, 2a. Whereas lectin pathway, let us come to the lectin pathway. How it is initiated? It is majorly antibody mediated. It is part of the innate immunity. It is not forming the antigen antibody complexes directly. It is attaching to the antibody. The antibody is attached by what part of this is the lectin. So it is called lectin pathway or MBL pathway. MBL means mannose binding lectin. So this mannose binding lectin attaches to the mannose residues on the microbial surface and gets activated. This is the initiation step. Here only one thing difference between classical pathway and lectin pathway is C1 is absent in lectin, in which place we have MBL MASP complex. MBL MASP complex means mannose binding lectin, which is bind to one protease, because it should have the protein lysing activity. So it binds to MBL associated serine protease. Serine protease is required. So this MBL, which can attach to the microbial surface, is attached with the proteinase, that is serine protease, MBL associated serine protease, to form a complex. This acts as C1, but it is not a component of a complement proteins. MBL MASP complex is not a complement, but it initiates the complement, whereas in C1, in classical pathway, C1 initiates the pathway, whereas in MBL pathway, MBL MASP complex initiates. But it should be remembered that it is not a component of the complement proteins. Okay, so starting complement protein of the lectin pathway is C4, whereas starting complement in the classical pathway is C1. So here, as the MBL MASP complex is same, remaining 4A will be attached, 4A will be released, 4B will be attached, and 2B will be released, 2A, which is active, will be attached. So instead of C1 4B 2A, the lectin pathway has MBL MASP 4B 2A, which is acts as a C3 convertase, right? So remaining all the same. Until C3 conversion, I am going to tell you. In this MBL MASP, if you compare, C1Q is similar to MBL, and C1RS are resembling this MASP, because MBL is the binding one and MASP is the converting C4 into C4A and B, right? Now coming to the alternate pathway. Alternate pathway, as I said, it is activated by antigen, especially the endotoxin, which is released by the gram negative. So if you see the alternate pathway, it is mainly concerned with the endotoxin major portion. Other activating proteins are also activating antigens are also present, but major thing is the endotoxin, which is the initiator for the alternate pathway. What are the others? You can see these are the different antigens which initiate the alternate pathway. And the major one is endotoxin. We have teichoic acid, fungus, yeasts, parasites, virus, even other than microorganisms like complex antibodies, heterologous RBCs, tumor cells, etc. So this will initiate the alternate pathway. So once alternate pathway directly acts on the C3, here also C1 is not present. C3 is activated to get C3b, and C3a is released. C3b, it is bound with the initiator, so it is called as C3b bound one. So which is attaching the antigens? It is the C3b, which is present in the circulation, which is attached with the initiators. Now this C3b bound one takes up the factor B, attaches with the factor B, then factor D comes and lyses this B, factor B, to BA and BB. So factor D is not seen here. It is only an enzyme which lyses the B, which is already attached to the C3b bound one. So here, what will be the C3 convertase? C3b, BB is the C3 convertase. So what is the C3 convertase in the alternate pathway? It is C3b, which is the bound one, and B is the factor B, in which factor BB is the active form, BA is released. But this C3b BB complex C3 convertase is very delicate. It has half-life of only 5 minutes. So what is required is this propyridine. This stabilizes this C3 convertase of the alternate pathway so that it can stay till 20 to 30 minutes. So propyridine acts as a stabilizing agent. So for the initiation of alternate pathway, what are required? C3 is required, factor B is required, factor D is required, and also propyridine is also required. 4 factors are required. Whereas classical pathway, what are required? C1, C4, C2, these are required. For lectin pathway, MBL, MASP, C4 and C2, these are required to form the C3 convertase in different pathways. So ultimately tell me what is the C3 convertase of classical pathway? It is C1,4B2A. And what is the important C3 convertase in a lectin pathway? It is MBL, MASP, C4B2A. And in alternate pathway, it is C3B, which is the C3 convertase. Once you know C3 convertase, what does it do? It converts C3 into C3B and C3A, which is released. C3A is released and C3B binds. So what is the C5 convertase here? This is the C5 convertase of different pathways. So for this we are adding 3B, that is C1,4B2A 3B, and for this C3 convertase you are adding 3B. So it is 4B2A 3B, and for alternate pathway C3 convertase we are adding 3B. So it is 3B, BB, 3B. That is the C5 convertase for different pathways. It is easy. Once you know C3 convertase, you can write the C5 convertase. And what is this all are doing? They are converting C5. C5 to active C5B, and C5A is released, which attaches to 6, 7 and also 8, 9, which forms a membrane attack complex like a tube. It will be placed into the cell membrane, so that it has inside this hydrophilic end and outside hydrophobic end. Whatever the material, water enters through this tube and the cell becomes bulged and get lysed. They form pores. This membrane attack complex forms pores over the surface so that it gets lysed. That is how the complement pathway is occurring and the action is taking place. So main thing, remember the initiator and the C3 convertase. What is the initiator for the classical pathway? What is the C3 convertase? In this way, for all the pathways, we have to remember C3 convertase and the initiators, right? So next, tell me the differences between these pathways. What is the difference? Which is the starting point? That is initiated for classical pathway, it is antigen antibody complex. For the alternate pathway, these are antigens, majorly endotoxin and others like even teichoic acid, yeast cells, these virus cells, and these all, other than that, cobra venoms, RBCs, these are all the alternate pathway activators. Coming to lectin pathway activators, it is the that is mannose, it is MBL. Lectin is attached to the mannose. Wherever mannose binding protein is present, that carbohydrate is attached to this lectin antigen, that will be the initiator. Now first complement activated, as in the classical pathway C1. No other pathways have C1, only classical pathway has C1. Alternate pathway starts with C3b. We know that C3b is binding to this of any of this antigen to become a bound C3b. And the lectin pathway, it is starting not with C1, it is starting with MBL, but here the first complement activated is the C4. It is MBL is not a complement. And you know C3 convertase is 1,4,2 C3bb and 4B2A for different pathways. When 3B is added, it becomes C5 convertase. And what complements levels will be low in the serum? All will C1 to C9 is utilized in the classical pathway. When classical pathway is activated by the antigen antibody complex, it utilizes all the components C1 to C9. Whereas alternate pathway, it is not using C1, C4 to C2. Only 3B and B and D, these are utilized. So this C1, C4, C2 will be normal in the alternate pathway, whereas in lectin pathway only C1 is not utilized, remaining all are similar to the classical pathway. So C1 will be normal, others will become low. So classical pathway is a part of acquired, as I said initially, alternate and lectin pathway or innate immunity. So this is the important, if you will at least remember this C3 convertase, it is an initiators, it is more than enough to write the pathways. Very easy if you remember that. So what are the effects of effective functions of the complement? Once the MAC, that is membrane attack complex, is formed, then it lyses the target. You know how it lyses the target cells. It forms spore. This complement membrane attack complex forms spores over the cell and it lyses the cell. And what about the inflammatory response? As we know, this is releasing the anaphylactotoxins and opsonization and the increasing the phagocytosis. The antigen antibody clearances in the blood is done by the complements, and the virus neutralization is also done. So phagocytosis is increased because it is releasing the opsonins, and the inflammatory response is increased because of the complements, and the immune complexes are being removed and the viral neutralization is being done. These are but the major thing is the MAC complex, which attacks the target cell to get lysed. That is a major component. So we know about the pathways and its effects of the complements. Let us see the complement receptors. As I said, complement is going to attach to the with the certain cells, so that cells should have some receptors. Which part of the cell will receptor is attaching to this we need not remember much, because whatever the receptors which are present, it will be named as CR1 to 5. CR1 is the major complement receptor. That if you can remember, it is enough. For theory exam, it is not so important. Coming to the evasion mechanisms of the complement system by the microorganisms, because these complements will kill the microorganism because they are forming the pathways and killing the target cells. So to prevent that, the microorganisms have certain mechanisms, like Escherichia coli and Salmonella has long polysaccharide chain, so that it prevents the MAC insertion. Neisseria gonorrhea also has the covalent interaction, so that it cannot accommodate the MAC. And the Pseudomonas, it will destroy CPA and C5A, which are the opsonins, which initiate the phagocytosis or increase the inflammatory response. And Staphylococcus and Streptococcus also prevents the MAC insertion. Pneumococci, C3b, and vaccine viruses majorly they release protein which resemble complement regulatory proteins. So what are these complement regulatory proteins that we need to import? This is important part in the complement. How the complement pathways are regulated, because if regulation is not present, the complement will act on their own. So there will not be any restriction. Majorly the activation is done by the microbes or microbial parts, but inhibition is mainly present in the regulatory proteins, or else if it is continuously without inhibition, the pathway is being activated, it damages our own cells also. That is why we need the regulation. The first regulation is C1 inhibitor, which where is C1 present in which pathway? It is present only in classical pathway. So affected pathway is only classical. It inhibits the action. And next one you should remember is the factor I and factor H. Where is this I? Okay. Factor H, these are I and H, these are important in the complement pathways, which are in blocking the formation of C3 convertase and by binding to C3b, and in this it is present. Okay, see, factor I is present in all the three pathways. It is acting on all the three pathways. It cleaves C4b or 3b by binding to C4b. This is important factor, regulatory protein among this. And some regulatory proteins which are acting on the MAC, membrane attack complexes, like membrane inhibitor or reactive lysis, rest protein and homologous restriction factors. These factors are acting on the membrane attack formation, so it inhibit the MAC formation, whereas these factor I, factor H, DAF, these are restricting the C3 convertase, and the only first initiator is the C1 esterase, which C1 inhibitor, which is acting only on the C1 regulator, remaining on C3 convertase and on the MAC complex. So that is the regulation proteins. If you remember these names and which part it is acting, it will be sufficient. You can write off these things. Factor IH for C3 convertase, DAF also, and membrane inhibitor of reactive lysis, homologous restriction, and S protein for MAC formation regulation. So last important one is the deficiencies. When complement deficiency may occur when there may be complement protein deficiency or complement regulator deficiency. There are two types of deficiencies. One is complement protein deficiency and complement regulator deficiency. When complement protein deficient is present, the pathways will be inhibited at one stage. Whereas if we complement regulated proteins are deficient, what happens? The complement becomes more and more active without any restriction. That is how we have to remember the deficiencies. So among the protein deficiencies, the first factors like C1, C2, 3, C3, 4, which are present for the initiation on the C3 convertase forming, this, if it is deficient, what happens? It cannot form the C3 and C3 convertase, and the pathway will be stopped there itself, and no membrane attack complex is formed. So it results in diseases like the pyogenic infection. Because immunity is not present, the complement pathway is not acting, what happens? Whatever enters into the blood, these will not be killed, because the complement are mainly present in the blood, which will stop the infections present. Whatever the bacteria, parasites, whatever it comes, it is killed by the complements. So if it is not working, it leads to pyogenic infections, SLE and glomerulonephritis. If the propyridine and factor D, where are these present? Propyridine is stabilizing C3 convertase, and factor D is necessary for factor B conversion. These are present in alternate pathway. Alternate pathway, as I said, it is activating by the endotoxin, which are major part of the initiators. So which organisms release endotoxin? Majorly the gram-negative organisms. So usually the gram-negative like Neisseria and pyogenic infections occur if this pathway is restricted. And if a membrane attack complex itself, like a C5 to C9, is completely deficient, what happens? No pathways at all. Especially disseminated infection, especially disseminated Neisseria infection is common in this deficiency. So the first parts, if it is deficient, it is leading to glomerulonephritis, pyogenic infection. If alternate pathway factors like factor D and propyridine are deficient, it leads to Neisseria and pyogenic infections, and membrane attack complex deficient, it is leading to disseminated Neisseria infection. So these are the complement protein deficiency. Coming to the complement regulatory protein, if it is not regulated, that is inhibition is stopped, what happens? It gives to overactivity. So overactivity or deregulated functions occurs. Mainly this are the important, as I say, C1 esterase is only for classical pathway. So if it is inhibited, inhibitor is inhibited, it leads to overactive classical pathway, leading to edema, hereditary angioneurotic edema, which is a serious condition. Hereditary angioneurotic edema, which is important condition. And decay accelerating factor or CD59, as I said, it is present, it is in regulating the MAC complex. So MAC complex, if it is deregulated, what happens? the increased lysis of all the cells, even our own RBCs will be lysed, leading to hemoglobinuria, paroxysmal nocturnal hemoglobinuria, because MAC complex is overactive here because of this inhibition of the regulation of the MAC by the decay accelerating factor. And factor 1 and H, these are regulators for which part? It is the C3 convertase. So C3 over consumption occurs, so immune complex diseases and recurrent pyogenic infections are common in this regulator deficiency. So basing on the action of the regulator, the diseases and the action occurs in the organisms. As I said, C1 is for the classical pathway, only classical pathway will be active, and it leads to angioneuritic edema. Decay activating factor is only for the MAC complex, so RBC is also lysed. And I and H, C3 convertase, immune complexes are diseases maximum. This is about the deficiency, complement deficiency. Regulatory protein and complementary protein deficiencies are present, and complementary regulatory protein deficiency. Whenever the question asks like name complement deficiency disorders, then you have to write both complement protein deficiency and complement regulatory protein deficiency also. So now you know the different pathways, like a classical pathway, lectin or MBL pathway, and the alternate pathway. And you know the complement deficiency disorders. First we have learned about the regulators, then we have learned about the regulatory protein deficiencies also. So shall I go to the questionnaire? Answer these 5 questions only. What is the C3 convertase of classical pathway, C3 convertase of alternate pathway, C3 convertase of lectin pathway? Angioneurotic edema is seen in the deficiency of. Disseminated Neisseria infection is seen in the deficiency of. These are commonly asked questions, either in the multiple choice, PG entrance, or in the Viva's. We have to remember C3 convertase for all, so I have given the C3 convertase, so that if it is remembered, you can write off the pathways. Answer, I am going to give 5 minutes to you. Within 5 minutes you have to answer these questions. Only 5 questions. Vrishni, please check once again. Okay, time up. The answers for C3 convertase of classical pathway, it is C1, C4b, C2a. But remember, if while writing in the exam, you should write a line over it, because it indicates these are active. When you are writing a line over this C1, C4a, C3b, you have to write on which the line should be present. It is not possible in the while typing, but you can write in the exam. C3 convertase of alternate pathway, it is C3b, BB, and C3 convertase of lectin pathway, it is MBL, MASP, C4a, C4b, 2a. And angina neurotic edema is seen in the deficiency of majorly the classical pathway deficiency, that classical pathway activation, overactivation, that is by C1 esterase, C1 inhibitor deficiency. Now disseminated Neisseria infection is seen in the case of majorly disseminated, means it is the major part affected is the MAC, C5 to 9. Okay, thank you all for answering. Meet you in the next class. Thank you.